And let me just start by thanking our today's speaker, Dr. Javier de Felipe. I think Javier doesn't need to be introduced to you, but let me just say a few words. Javier is a doctor in biology and a PhD by the Universidad Complutense de Madrid, where he started his research on the cerebral cortex. He then moved to the States to work at Washington University and also at the University of California at Irvine. And in the 1991, he came back to Madrid to get into the Instituto Cajal of CSIC, where he formed a research team to analyze alterations in the cerebral cortex in patients with epilepsy originally. In 1997, he participated also in the NASA NeuroLab project to study the impact of spaceflight on neural circuits in the brain. And in 2006, he began researching the effects of Alzheimer's disease on the microstructure and microorganization of the cerebral cortex. He's the director of the, or has been the director of the Centro de Investigación Biomédica in Madrid, at the Universidad Politécnica, and also the Cajal Laboratory of Cortical Circuits. He has participated in the Blue Brain Project and later on the Human Brain Project, that many of you may know of. And he's been a recipient of a number of national and international awards. And I think it's a very well-known researcher and a big representative of CIBERNED. So the title of his talk today is Alteration of Cortical Circuits in Alzheimer's Disease, Implications in Cognition. So Javier, thank you very much for coming, for your willingness to share your work with us. And you can please, yes, share the screen and go ahead. So welcome and thank you very much.
Thank you. Can you see the presentation?
Yes, yes.
Okay, very good. Okay, first of all, thank you very much for this nice introduction. Hello, everyone. So today, I'm going to talk about alteration of cortical circuits in Alzheimer's disease and implication in cognition. So I'm going to make an introduction, then I will talk about promising technologies to study the microorganization of the brain, and then one of the conclusions is that neural excitation and inhibition imbalance as the endpoint of all Alzheimer's disease and related micro-level synaptic pathology. So, as you well know, there are two major hallmarks in Alzheimer's. One is the extracellular fibrillar amyloid beta peptide that are accumulated in the brain, and then the intraneural neurofibrillary tangles consisting of aggregated phosphorylated tau. So this is a progressive accumulation of this pathological aspect. These are all of you know very well this. So there is a progression from the temporal lobe towards the other regions of the brain, and there is this progressive alteration of pathology is correlated with the progressive loss of memory and deterioration of behavioral and cognitive function, as you can appreciate in these drawings of William Hunter model, which is reproducing very well how the brain probably is the changes that this painter was suffering when he had Alzheimer. So also all of you know very well that for unknown reasons, there are, for many years, you can have beta amyloid and tau. However, this is asymptomatic and impossible diagnosis. And then after a few years, then you can develop mild cognitive impairment and then dementia. So from the very beginning, in my laboratory, we were wondering what alterations in the synaptic circuitry explain the early cognitive decline in Alzheimer's disease and how we can we prevent these changes, and why it's so hard to answer this question. Before I continue, I would like to emphasize that that in our laboratory, we are analyzing also the normal human brains, and it's very common to find this kind of pathologies in normal people. I mean, control people that don't have any kind of deterioration, something that also is very well known. But when you see in your hands, when you see in the preparations, patients with Alzheimer and others that are with similar pathologies, at least at the early stage, is quite interesting and difficult to understand. So why is it so hard to answer this question? Why there are some cases that you can see these pathologies and there are no deterioration at the early stage, because at the later stage is impossible. I mean, it's very easy to understand that these people, they have a lot of damage. Right? So a major problem is that both beta amyloid and tau can affect neural activity at multiple levels of complexity, from molecules, synapses, neurons, circuits, and networks. And it's very difficult to integrate all of this information to have a general picture. But this is not impossible. This is a major question in neuroscience, is what can be done with data, how can it be interpreted? And that is Sidney Brenner, that was a Nobel awarded in 2002. He said, we are in a sea of data and starving of knowledge. The biological science has exploded, largely through an unprecedented power of accumulated descriptive facts. We need to turn data into knowledge, and we need a framework to do it. So in other words, we need to transform the data in knowledge. And that is quite difficult. For those reasons, big projects have been developed to try to how we can integrate multiple data, multi-level data, to better understand the alterations. In general, the basic circuit of the brain is this. You have the input, the output, you have the oxygen and nutrients, and you have projection cells, which are in the cortex are the pyramidal neurons, interneurons, most of them GABAergic, glial cells, where you have three major types, astrocytes, oligodendrocytes, and microglia, and the neuropil, which is the region between the neurons, glia, and blood vessels. However, the magnitude of the problem is huge to understand the human brain, because the brain works as a whole, and thousands of regions and subregions can be identified within the brain based on the morphology, physiology, and molecular and connectivity of the neurons that make up these regions. But data and knowledge is fragmented, and how the brain works as a combined unit remains a mystery. So this is the major problem. We know a lot of issues about experimental animals, but not in the human brain. This is a major difficulty. However, we will see that there are now methods that we can apply directly to study the human brain. So there are three levels to analyze the brain, the human brain. One is the macroscopically. You can track the connectivity through diffusion, tensor imaging, intermediate resolution with light microscopy, where you can analyze the putative contacts, and then the ultrastructural level map of the synapse, which is the most difficult level of analysis of the human brain and any other species. We are going to today focus on this intermediate resolution and the ultrastructural level, and emphasizing that this can be analyzed even in the human brain, which is critical to better understand what's happening in Alzheimer. No? So pyramidal cells are the most common neuron. They represent about 70 to 80% of the total population. They represent the majority of projection cells, and they are located in rural areas. And they are the major source of intrinsic connectivity, of sensory connections, and the spines are the major target of synapses in the brain. So also it's critical to to keep in mind that pyramidal cells are responsible for the spread of activity through the action collaterals. So this spread of the pathology or the connectivity of functional organization depends very much on understanding the principles that determine the geometry of pyramidal cells, which is essential to comprehend how neural circuits operate in the cerebral cortex. So now the idea is that we can obtain data from the neurons, data about the morphology, molecules, etc. But we need models to understand the data, to convert, to transform this data in knowledge. So in analogy to electronic circuits, neurons correspond to electronic parts, and neuron cells types correspond to what kind of parts are they. So the classification of neurons are not only important for the practical point of view, because when we talk about between us and talking about Martinez cells, basket cells, etc., we need to give names, but also because they have different functions. And so therefore it's very relevant for the functional point of view. So the idea is now is how you get the data on the real neuron, you visualize or label the real neurons, and then you transform these neurons with using computational mathematics and computational neuroscience in virtual neurons that to allow you later to do models, simulations, to understand which are the different parameters. And to do this, as I explained in several other occasions, a good thing is that you can obtain fantastic neurons in the human because it is feasible to use intracellular injections in fixed human cortical tissue. That means that you can use tissue from autopsy cases from the human. So you can see here, this is the cerebral cortex, this is layer 1, different layers, and then with a microelectrode you can go and inject the neurons using a confocal fluorescent microscope and microelectrode. You can penetrate within the neurons that are labeled with DAPI, and then you label with a fluorescent marker. In this case, is Lucifer gel, but you can use other kind of markers. So you can visualize the detailed morphology of neurons of the human with an extraordinary quality. Here we can see these spines that are called learning spines because are dynamic and are thought that they are involved in the production, in the new contacts. When we learn something, we establish a new circuits. These other, which are with a big heads, are thought to be memory spines because are stable and are thought to be the trace of our memories. And also, it's important, and we will see in a second, the morphology of the spines because are related with the functional activity, I mean, with the functional synaptic properties. So these intracellular injections of cortical tissue, you can inject in multiple regions of the temple, in this case, the temporal lobe, and then using these techniques, we have recently published an article in which, for the first time, you can visualize the neurons of the human with a very high quality that allow you then to transform this data in virtual neurons and to include the data in models, as we will see later. So the morphological parameters are critical because these are properties that are related to the functional properties. And we know that the pyramidal cells with bigger arbors can sample a bigger portion of the cortex than those that have smaller arbors. The complexity also is related to the functional properties of the neurons. So it's critical to understand the variations of the different pyramidal neurons, even within the same region, because they are in the CA1, as we will see in a second. There are variations in the morphology. So we need to know which are the range of variability and of the morphology of the neurons that are in each part of the brain. The The spine, the morphology of the spine directly reflects its function. Almost all the spines establish a synapse, so changes or differences in the number of spines in the dendritic arbor neuron may influence the cerebral system cortical functions. So the spine volume is correlated with the area of the postsynaptic density. The length of the spine neck is proportional to the degree of biochemical and electrical isolation of the spine from the parent dendrite. Larger spines can generate greater synaptic currents than smaller spines, and these spines are dynamic structures with fluctuations in volume that seems to have an important implication to cognition and memory. So therefore, we have a very complex neuron. The spines, which are not just simple protrusion of the dendrite, these are very complex structures, and these spines, some authors consider the spines are the unit of information instead of the whole neuron. And this is just to have an idea. There have been identified more than 1,000 types of proteins in the postsynaptic densities of in the dendritic spines. So this is quite complex. The study of the dendritic morphology is critical then for the study of from a functional point of view. So now we have a fantastic method to visualize the neurons, but then we need tools to analyze the numbers and density of the spines. Before, a few years ago, the way to do this is you estimate the number of spines in a given region of the dendrite, and then you make an estimation, making a calculation of the whole dendritic arbor, and then multiply by the density found in a given region. Now there are tools that allow you to label practically very fast all the spines, so you don't need to now to make estimation because you now visualize all of them practically. So the other issue that we need is to have the data about the morphology, not only the density and the number, but also the variations in the morphology, because as I say, the morphology is directly correlated with the functional synaptic strength of the inputs of each spine. This software is not yet available, and we use is manually do this using other software, but it's not automatically. We are trying to develop a tool using machine learning and other deep learning to do this automatically. But still, we have not been able to do it. But this is the next thing that we would like to do. So now, the dream is to have the map of the synaptic strength of each synapse established on the dendritic tree, is to just to see what is the synaptic current generated by each spine in the neuron. So this is the dream. So then at the electron microscope level, we have we need to know the density and number of oscillatory and inhibitory synapses, the spatial distribution of the synapse, the size and shape of the active zone of the presynaptic terminal and of the postsynaptic density, the identification of the synaptic targets, for example, there are the dendritic spine, the dendritic subs, cell bodies, etc., and the ratio of the density of synapse and mitochondria to model energy consumption. So these are the things that we need to know. So now we have a very good machine that is called this is working now. You see? Yes. So it's a FIB-SEN that is a machine that combines a high-resolution field emission scanning electron microscope column with a focused gallium ion beam, which permits the material to be removed from the sample surface on a nanometer scale. So you can see here you can go with a gallium, and you can go nanometer by nanometer. We usually put this sputtering is about 20 nanometers, and you can include the whole hippocampus, like in this case of the human. These are the preparations, and these are the samples. So you can sample regions in the dental gyrus, CA1, CA2, and you can identify the all the synapse and to analyze in 3D, something that is amazing because this kind of information is not available in the human. And we have developed over the years methods to analyze this in autopsy cases, like this is an autopsy case, which is the quality you can see that is very good. We will talk later if you want to see this. So now we have also a very nice method to obtain 3D reconstruction of the brain, but now we need a method to visualize the synapse and to extract the information. So we developed a method, an informatics tool that we call the Spina, where you can see in the stack of images, you can label all the synapses that are excitatory, which are in green here, and the inhibitory in red. So you obtain the real number of synapses. We don't need to make an estimation of how many synapses are there. It's just only you count the absolute number of synapses. And here you can see also all each synapse. You have the data about the morphological parameters like the size, etc. And then you can go, you can see here, back and forth. You can identify the postsynaptic elements and many other features. So all this information is obtained and you can extract and then to use for computational models. So this is from a paper that we have recently published. This is the human C1, where we have analyzed all layers. These are the 3D reconstructions, and these are the data about the targets, the number of synapses on the spines, the head, the neck, etc. So this data was available more or less, but not very much in animal models, in rats and mouse. But in the human, we can obtain this data even now in more detail because this has been obtained from a serial section of thousands of synapses. So the idea of this is you have the integration of data you have from the cellular components, the morphological parameters. You analyze individual cells, the parameters that are important to better understand the functional organization of individual neurons, the map the synaptic currents based on the morphology of the spines, and then electron microscopy, where you can use 3D automatic electron microscope to analyze the postsynaptic targets and other parameters of the synapse. And in 2015, we published a paper that was with a blue brain. We were 85 authors that represent the study for several years. We collaborate with the laboratory of Henry Macan and other laboratories around Europe and to propose the first draft of the reconstruction of cortical circuit, and this is the rat neocortex. So here we include the neural physiology, neural anatomy, synaptic physiology, and this was involved computational neuroscientists, engineer physiologists, anatomists, etc. So this is a simulation of a column based on this data. We say that this is a drug because here has not been included yet the glia or the blood vessels or the spines. So this is based on a lot of data about the different types of neurons that are known so far, morphology of pyramidal neurons, etc. But the important thing is that this study, this project, sorry, it's not working now. Well, anyway, the important thing is that we have here the pipeline to create this activity. So now the thing that we are doing is to add new information. For example, we are going now to introduce spines and many other features. So in a near future, we hope to have a more detailed, a realistic model of the piece of the neocortex. This is the idea. And also we are using this because imagine that, for example, we would like to know what is the effect if you decrease or increase a given parameter like a receptor or a neurotransmitter, or block a particular type of neuron, whatever. So the idea of this is to try to understand which are the functions of the participation of the many different types of neurons that are in a given column. So when talking about the application of these methods to study the Alzheimer disease, this is an important aspect is that the beta amyloid is deposited in the brain, but you can see that these beta amyloid, some of the beta amyloid plaques are in the neuropil and others are close to the cell bodies. And this is about 60% are in contact with neurons and 40% in the neuropil. And the impact of this is important because this is the innervation of GABAergic and glutamatergic terminals in the cortex. And when you have the deposit of beta amyloid, what is the influence when you consider these kind of characteristics? No? So, and the membrane surface of neurons in contact with plaques are devoid of GABAergic terminals. This is an important feature because here you see this is a bugat, the label here, this is a plaque. And you can see these neurons that are close to the plaque, higher magnification. You can see this region here that are devoid of GABAergic terminals. So this decrease of GABAergic input on the soma. So I will talk about that later. So another thing that we were doing is to analyze what happened to the dendrites that are in contact with plaques. So to do that, we inject neurons near the plaques, and to do this, we did intracellular injection, like this. So and then we stain for tau in this case. And you can see here, this is a neuron that has been is tau negative, and this is tau positive. So you can compare between tau positive, and this is using 88 antibody, and then we compare neurons that are positive and negative, and we analyze the spines, and you can see here. So this is a neuron that is positive. You can see here at the high magnification. You can see the spines here, the dendrites. You can see a clear decrease in the number of spines. But this decrease doesn't occur in the whole neuron. This occurs in parts of the dendrites, and then progressively is decreasing these spines. So this is what happened when it's in contact with the beta amyloid. Sorry, there was misplaced in this slide. That should be before. So here you can see there's a dendrite that is in contact with a beta amyloid plaque. And you can see here a clear decrease in the number of spines, but not here. So this is like a mutilation. So because the alteration is local in the case of the spines here. However, when you do intracellular injections in neurons which are tau positive, these are alteration of the spines affected to the whole neuron. So, but this is an important feature that we should consider. Beta amyloid, what is in the neuropil, is affecting locally some of the spines, but the rest of the spines of the neurons are practically intact. So this is again neuron. This is a neuron with intracellular tangles here, and you can see here in this case the spines. You see there's a clear decrease in the number of spines. Even you don't need to count the number of spines because it's a clear alteration. So the accumulation of diffuse phospho-tau in a pretangled state does not induce the dendritic change, whereas the presence of tau aggregates forming intracellular interneural neurofibril tangles give rise to a progressive dendritic atrophy and loss of dendritic spines, asynaptic disconnection. So in summary, you have a beta amyloid-induced local alterations on the dendrites and soma in contact with amyloid. Loss of local dendritic spines and loss of perisomatic GABAergic terminals. And this will give rise to hyperactive neurons, epileptic form activity that we will see, I will show you in a second. And the presence of tau leads to a progressive dendritic atrophy and the loss of dendritic spines. And this connection is probably related to the cognitive deterioration. However, the other, the alteration of the given around the perisomatic GABAergic terminal probably give rise to a neural excitation inhibition imbalance because the local circuit is an imbalance of these GABAergic inputs. So, in summary, you have this innervation of the pyramidal neurons. These are different types of interneurons. Some of them innervate the apical dendrite, which are different types. I'm not going to enter into details. There are other neurons like the basket cell Alexander cells, which are responsible for the perisomatic innervation. So these are critical for the control of the activity of pyramidal neuron. However, these other interneurons that are connecting with the distal portion of the apical or the dendrites are probably involved in plasticity and other mechanisms. But these basket and central cells are critical for the control of the activity. So if you in Alzheimer, what happened here is the same. It's not the same if you have an accumulation of the spine. Sorry, it would be like this. If you have a plaque in this region, the functional significance of the influence on the pyramidal neuron is going to be different if you have the plaque near the cell body. And as I say, about 60% of the plaques are in the neural pil, so will be here, and others are in contact. So now, the important thing now is we have a very complex or synaptic organization, of course, and the changes that occur are very variable and depends not only on the density of plaques that are present in the brain, but also which are the physical localization of these plaques. So it's not, as I say, for example, in Alzheimer, you will have plaques here and then here and here. And in summary, to try to understand which is the influence of the alterations of the circuits in Alzheimer, is critical to have models and to try to analyze what is the functional implication of these alterations in particular cells. So then we go to the macroscopic level. We have these levels. You have macroscopic, and this is the mesoscopic level. So what is the influence of the alteration of cortical circuits in the functional organization of the brain? This is again very difficult. If we go to the particular brain region, like I say in the cortex, maybe we can be closer to try to better understand the disease. But at the macroscopic level, we can use models like this from microscale, mesoscale, and macroscale, using the connectome maps, which are not very detailed. And then from the mesoscale, the imbalance of the dysfunction of neural circuits, which is due to this deposit of this alteration of the GABAergic control of the connectivity of pyramidal cells that gives rise to an alteration of the general connectivity. And then you have the decrease in the number of spines, which are involved in the excitatory inputs, which are losing, but not only that, but also in cognition, because the memories and things like that are may depend on the number of neurons with tangles, whereas the GABAergic, the beta amyloid is more involved to this imbalance of the GABAergic circuits. So, as I said, this is very theoretically. For example, now these models are using tractographies, which are not really the connections. And this is connected, A is connected with B, but as I showed you, the real thing is much more complex. And here, the models to use in this kind of macroscopic models are based on point neurons, which are not realistic neurons. So the one thing that we are proposing now in the human brain and also in the blue brain is to focus on the human temporal cortex in the hippocampus, sorry, because the connectivity is really is very well known. Practically, it's the same like in the macaque, with some differences, but it's quite well conserved. And we have now, we know that there is a progressive accumulation of tau, beta amyloid, as I say, to understand how the alteration affect the cognition and that disease. We need to make models to try to understand how these alterations affect the functions. And because we know all of these connections, and we have the morphology of the neurons in that particular region, we have the real details, and soon we will have the rest of the data on the morphology of different types of neurons in the human. And as I said before also, we have also at the level of the synapse, we have all the information. So now we can generate very realistic models to try to understand this alteration. Even now, for example, we have used this microscope, the FIB-SEN, to go inside the plaques and to analyze around the plaques, as you can see here, which are the changes in the synapse. So, in my opinion, I am very optimistic if we are using this kind of models to try to understand what is the influence of the alteration of the circuit. I think that it's difficult, of course, because just we are at the very beginning, the analysis, to have detailed models to understand the brain circuits. But the good thing is that we have tools to analyze the microanatomy of the human brain, and we have also the tools to develop computational models that I believe I think in the near future we will be able to make very realistic models of the human temporal lobe and to see what is the effect of the progress of the disease. And then we will go to the rest of the brain. But I believe that a good strategy, something that I always proposing and discussing, is to focus on the hippocampal formation of the human and then go with other researchers. There are many laboratories which are excellent researchers in using the human brain, physiology, anatomy, molecular biology, biopsies, everything. And I believe that we go together and analyze the temporal lobe, hippocampal formation in both normal and in Alzheimer. I think that we will advance very much, including MRI, tractographies, everything together, from microscopic, mesoscopic, and electron microscopic level, that we have all three levels of analysis focus on a particular region of the brain. And I think this is all that I want to say today. Thank you.
All right, hello? Thank you very much, Javier.
You have to hear me, right?
Yes, that's fine.
I thought I was talking, nobody was there.
I was fine. So thank you very much for a wonderful presentation. So now the paper is open for discussion. So anyone who is interested in posting a questions, please do so by raising your hand. or by send me a message through the chat option. So we already have a first question coming from Ramon Trullas. Ramon, please go ahead.
Oh, Ramon, how are you?
Hi, all right. Thank you very much, Javier. Extremely impressive, wonderful presentation. I'm really impressed. And I think it's extremely interesting, especially the idea or the results that you present that a plaque has a local effect in contrast with the PHF tau.
Yes.
And I wonder if you have data on the relationship of both. Are there any PHF tau neurons that do not have any contact with plaques?
Oh, well, yeah, this is a good question. My impression is that when you have plaques, it's more or less randomly distributed, you see. So you see preparation of the human with stain with beta-amyloid, and you label then the neurons to visualize dendrites, something like that. You see plaques that are in the neural pillar very close to the cell body. In a section, you can see several plaques close to the cell bodies, other only in the neural pillar. Some layers you have more in the dendrites, other more in the. So these are a large variability, a large variation in the pathological aspect of the beta-amyloid. But even within the same individual. For that reason, I said that it's critical to use models to try to understand what is the impact of this, because otherwise it will be impossible to understand. No? So, and then you have beta-amyloid. So you have sometimes beta-amyloid in a region with very few neurons that have tangles. And some regions you have a lot of neurons with tangles and very few plaques. So, and other regions that you have both. You see, so the idea at the end is to see what happened in the what you were asking when you have tau and then you have a plaque near this neuron, the effect will be stronger, of course, yes. But the important thing also is something that we have to keep in mind, that you have regions with no or very few plaques, another with very few tau on both. So the emphasis of this is that when I try to publish a paper with Alzheimer, we use usually 3 or 4 cases just to analyze particular aspects. And always the referees told us that there are N is very few, there are only 4. And I always say that 4 is always, N is 1, always. If you have 1,000 patients, you are going to have 1,000 ones, you know, because each one is going to be different to the other. So we don't need to analyze thousands of individuals to analyze this detailed aspect. We need more to have a approach, more mathematical approach to the problem, to try to understand what is happening here. You know what I mean? Is to just to analyze in detail particular circuits, and then to try to understand what is the significance of these alterations, not just to see what is the variation between different patients and so on, because as I say, there are not 2 patients equal, always are different, always.
Yeah, I also see the same. Extremely interesting, Javier, extremely interesting. And if there is no other question, if I may ask another one, do you see any relation, different morphology in the neurons that do not have plaques around and have the PHF?
Well, yeah. Well, this is also a very interesting thing. That something that we see. Well, it's very nice because sometimes you see a dendrite that when it's in contact with the plaque, start to have strange things, start to grow like a strange processes, or reduction of using the dendrite, start to make it very thin. It is very, very interesting, because imagine that when you go, the distal dendrite of this particular dendrite that is in contact with the spine is completely normal. So if you start to think what is the influence of the activity, so this particular neuron, if this is receiving information from that particular dendrite, cannot be normal because it has been altered. However, you get inputs from other regions of the neuron which are not in contact with the spines, sorry, with the plaques, are normal in that sense. So the same neuron may have may behave like a normal for particular connections and a normal for another connections, because you have one, I don't know, you can see me, you have like dendrite, this is not good, and the other one is intact. If you receive information here, will be fine, but if you receive from here, will be altered, you know what I mean? So for that reason, I insist, although I am just simply anatomist, to understand the data, transform the data in knowledge, we need computational models to try to understand these kind of things that you are asking.
This is fascinating. Thank you very much, Javier. Extremely interesting, and showing that a neuron is also an individual in itself, and we have
yes.
architecture. Thank you very much, Javier.
Thank you to you.
All right, thank you, Ramon, and thank you, Javier. So we have also a question from Pablo Castillo. So please go ahead. You're mute.
Oh,
okay.
Yeah. Hey, how are you doing? Thank you very much, Javier, for this excellent presentation. Very nice. I enjoyed a lot. Not from my own experience, but from my have read, there's this notion that cognitive impairments in Alzheimer's kick in much earlier before you see any of these pathologies. So maybe related to this question from Ramon. So the question is, given the granularity that you're getting with your studies, I imagine that at some point you should be able to detect problems, anatomical issues, and coming from a functional angle, but you should be able to see or detect some anatomical problem way before you see any plaques or tangles.
Yeah.
Based on what people say.
Yeah.
So do you have any evidence, or have you specifically tried to answer this question?
Yes. That is it's a thing that we are all the time thinking about, because, as you say, so the thing that we are doing now is to use brain tissue from individuals that are neurologically and cognitively normal. They have a normal life, and then you go to the temporal lobe, and then in some regions you have tau, mostly, and so another some beta amyloid. So the idea is to go there and then nearby and analyze the synapse, something like that, to see what happened, you know. And that is, so the question is why, when you start to have a deterioration, because sometimes I call the pathologist and I say, you tell me that this individual is from a normal individual control, but I see a lot of tangles here sometimes. So, and then I say, please, would you please confirm or call to the family or whatever. And the other day, for example, we have a case, a fantastic case, that was a woman that she was, I think, 85 years old, something like that, and the brain, she has a lot of tangles and alterations. And they told me that she was a control case. So he called the family again and then was talking with her daughter. She said that her mother was living alone at home. She was reading a lot, working, cooking, everything. She was completely normal. But then when you see the real cases of Alzheimer with cognitive deterioration and you analyze the brain, is not the same. It's different. At the electron microscope level, for sure, you have synapses which are very rare. It's not only the numbers that disappear, it's the morphology, the synaptic proteins that start to be abnormal. And then we have detected that kind of things, but in later stages. So that maybe happen, these alterations occurs is, first of all, we don't know, but probably is the neuron that start to be to start developing these tangles in the, and then at the beginning probably this phosphorylation of tau maybe is not pathologic, but for any reason then becoming pathological, and then start to see the spines to be decrease the size, then for the neurons start to be to change the skeleton of the neuron. So probably is the neurons that become abnormal, and then are the synapse, probably. There are people that say that there are changes in synaptopathology, but these studies are based on data. The quality of the data is not very good. You see the papers are, you see the quality is very bad, you know. So
Javier, you raised a very interesting case, meaning, I'm not sure how often that is, but meaning you can have tangles and yet you don't see any clear cognitive deficit. So at least in that case, so how often is that? Because that would argue against some causality.
No, right? No, it's because when the cases, practically all cases that are older than 65 or 60 years old, I am already 70, so maybe I have some plaques already and tangles. So the thing is that you have, but if you don't have this, you see that Alzheimer and control with tau is quite different. You have neurons, for example, you have some of them like cluster neurons in the hippocampal formation in the CA1, for example. Then if you go to the temporal cortex, you see isolated neurons, one here and another very far away. Something that is very strange because are isolated neurons that appear little by little. So the impact of a neuron to be damaged probably is nothing because there are millions and millions of neurons. And in the hippocampus of these patients, or the individuals, sorry, they have maybe, so you can see it because you see some clusters here and another there. You see, and sometimes you see a lot. But when you see them with deterioration, is the neuropil also, there's a lot of stress, and then it's a mess. Even you don't need to know to tell you that somebody is demented or something because you can see in the preparation. It's very clear. But the most interesting thing for me is when you reach in this point, you know, when is you start to have this deterioration. And this is what we are trying to understand. And the same happened with epilepsy. No? You see alterations. So why you have epilepsy, other people don't have it, it's having the same damage. So we are trying to understand the alterations, but as I insist, although we produce beautiful data, very nice, it doesn't mean anything if you don't have a model to try to understand what you are seeing. Your questions can be answered only when we have a model to try to understand.
All right, thank you very much, Javier.
You're welcome.
All right, thank you, Pablo and Javier, for a nice discussion. So we have another question now coming from Carlos Matute. Carlos?
Only my friends are asking things.
Yes.
Beautiful thing.
So Javier, a beautiful work and also very relevant. Congratulations. So
Thank
I have two very specific questions. So in the two circumstances, either plaques or tau positive neurons, what kind of spines are most sensitive to damage? So, I mean, there are several different types, the thin, the stubby, mushroom, philopodia, whatever. And this is one question. And the other one is, how about when you have no tau hyperphosphorylated in the neurons or no a plaque? Because the understanding is that beta amyloid is in excess everywhere in the AD brain and it creates damage. So what happens in those neurons which are not tau positive and are not within the vicinity of a plaque? So thank you.
Yeah. Well, the thing, we published an article with mathematical experts in mathematics just to analyze the loss of neuron spines or in dendrites of these tau positive neurons. And they found that these loss are in clusters. When you have in clusters, you see, no, it's only one here, another there. It's 2 or 3 disappear, then another 2 or 3. So probably are spines that are connected with particular other type of neurons or whatever, you know. We don't know yet this. So also because we are analyzing a fixed tissue, the spines that we are seeing are those that appear at that particular time. So probably one spine that has a big head, a large head, maybe in 1 hour later is thinner or something. So the only thing that we can do is that we analyze the winged neurons and compare between neurons that are tau positive or tau negative within the same region, the same individual, and are neighbors. Otherwise, it will be very difficult to understand because different regions are different morphologies, different humans will be different. And we found, our controls are those that are tau negative, you know, so compared to the ones that are. So these studies are quite difficult because before you do the injection, you don't know if it's tau positive or not. So the thing that we have to do is to inject many neurons and then label for tau and see which of those are positive and which are negative. And if they are good intracellularly labeled, then it's fantastic because you can see, sometimes are beautiful because you see the neuron is injected and then they don't have almost dendrites. It's like an atrophy. Others are very full dendritic arbor, but with very few spines in the distal portion, something like that. So we are trying to understand what happens when you have tau or when you don't have tau. And when you have the plaques, all the dendrites are touching the plaques. They have suffered different changes, which are, as I said, the dendrites are thinner, the spines are disappear, or sometimes you see some spines that are very big, start to grow. And then outside the plaque, the same dendrite is completely normal. So it's very local, it's something amazing. It's something, and then when you think that you have a dendrite are touching plaques, another dendrite that does not touch the plaques, then the input arriving to the different regions of the neuron may work fine or not, depending on what kind of inputs are receiving in the, you know, so it's very nice. It's interesting, but we don't have any responses to see. It's beautiful, but we don't know.
Thank you.
All right, thank you, Carlos and Javier. So we have time for 1 or 2 more questions, if there are any. I don't see any raised hand, or I'm not receiving any message through the chat. So I wonder whether anyone else wants to pose another question. More questions? All right, so I don't see any messages coming through the chat, nor any raised hands. So I guess it's then time to close the session. But not before thanking once again Javier for a wonderful presentation and a very nice discussion, and his willingness to share his work with us. Thank you everyone for attending. And so we will have another webinar in 2 weeks' time, as usual. So you're all invited to attend as usual and always. And that's it. So no more questions coming. So we're gonna close here. So thank you, Javier. Thank you, everyone, and have a great day.
Thank you.
Bye bye.
Thank you, Javier.
Bye bye. Thank you.
Bye.